Good morning, sir. Manimurugan, sir. Good morning, sir.
Good morning. Good morning.
Okay, sir. Shall we start, sir?
Yes, we shall start.
Okay, sir. A very good morning to everyone, and I welcome all of you for the third day sessions of 10 days online national level training program on emerging trends in seed production technology and quality control, effective seed supply chain of horticulture crops. Today, on third day, we are having 3 lectures. In the morning session, we have a lecture on nuclear seed production and maintenance breeding and overview, which will be delivered by Dr. Manimurugan C, scientist, ICAR, Indian Institute of Oilseed Research, Hyderabad. And in the afternoon session, we will be having another 2 lectures. One will be on vegetable seed advances in vegetable seed production, and another one is on vegetable seed production under protected cultivation. Now, may I now invite Dr. Ganesh Gowda I. Patil, sir, to introduce Manimurugan C to the participants. Over to you, sir.
Thank you, Dr. Dhananjay, giving me an opportunity to introduce Dr. Manimurugan, sir. So I just read out the biodata of Dr. Manimurugan C, sir. Sir is working, Dr. Manimurugan C, sir is working as a scientist at Indian Institute of Oilseed Research, Rajendra Nagar, Hyderabad. And sir is specialized in the discipline of seed science and technologies. Sir has done his undergraduate and postgraduate degree program from Tamil Nadu Agriculture University, Coimbatore. And sir has also completed the PhD program from Indian Agriculture Research Institute, New Delhi. Sir is having a very expertise in the areas of seed storage and seed quality maintenance, and also like seed maintenance, the maintenance of breeding of vegetable varieties and hybrids. And sir is 10 years of very vast experience in the breeder seed and truthfully labeled seed production of vegetable varieties and hybrids at Indian Institute of Vegetable Research, Varanasi. And apart from this, sir is also expertise in the areas of seed quality testing. So apart from all this, sir is acted as a seed sale in charge at Indian Institute of Indian Institute of Vegetable Research, Varanasi. And sir also given more than 25 guest lectures in the national training programs conducted at National Seed Research and Training Center, Varanasi, during the year 2015 to 2020. And sir is also designed and conducted the many training programs at Indian Institute of Vegetable Research Institute, Varanasi, namely the principles and production techniques of hybrid seed in vegetables, and another one, the pre and post harvest management techniques for seed quality assurance in the year 2016 and 2017 respectively. So apart from this, sir has published many research articles in the very reputed journals. So more than 15 research articles published in the research journals. With this, I request Dr. Manimurugan C, sir, to give his valuable guest lecture on the topic of nuclear seed production and maintenance, breeding and overview. So I request Manimurugan, sir, please take a charge and continue the topic, sir. Thank you, sir.
Thank you, Dr. Dhananjay and Gowda, sir.
Thank you, Ganesh Gowda, sir, for introducing today's speaker. Sir, on behalf of University of Horticultural Sciences, Bagalkot and College of Horticulture, Bidar, we are welcoming you to deliver your lecture, sir. Over to you, sir. Mani, sir.
Thank you, Dr. Dhananjay. Should I share my screen?
Yes, sir. Yes, sir. Please, sir.
Is it visible now?
Yes, sir.
Am I audible?
Yes, yes, sir. It is very clear, sir.
Okay. Good morning to all. Today I am going to deliver a lecture on nuclear seed production and maintenance, breeding and overview. Actually, nuclear seed production and maintenance, breeding, both are one and the same. It involves breeder seed production also. If you see the importance of nuclear seed production as well as maintenance, breeding, in this slide, you can see some of the varieties. I didn't listed all the varieties of IAVR. Only some of the vegetable varieties. Tomato, the Kashi Vishesh, Kashi Aman, Kashi Abhiman. So some of the tomato vegetable varieties released during 2006, notified during 2006. Still, these varieties are in breeder seed production chain. Among the national indent, every year IAVR is receiving the indent for these varieties. So these varieties are also having a lot of demand among the farmers in northern plains of our Indian condition, UP, Bihar, in those districts, Madhya Pradesh also, those state, still a lot of demands are there for these varieties. So these varieties nowadays, many new varieties are also came, but still these old varieties are having demand, and these old varieties are having in seed production chain. It is only because of maintenance breeding. If we are not doing the maintenance breeding, it is very difficult. It is very difficult to maintain the trueness of the particular variety. So that maintenance, this is the importance of maintenance breeding. Only because of the maintenance breeding only, still these varieties are surviving. These varieties as well as hybrids are surviving in our country. If you see the scheme, that is variety development, seed production. If you see the scheme, first in any institute, national institute, state agriculture university, through the breeding program, breeders are developing many varieties, many improved cultivars. Every year they are developing based on the need. Then at the time of release of the variety, the breeder may have a very less quantity of nucleus seed in his hand. The nucleus seed is seeds of good variety. There is a handful of seed only, very less quantity of seed only. The collected from the variety or plant which is showing desirable characters as well as high yield. So from that, the breeder will select little quantity of seed. So after that, it will be identified and notified and released and notified. So during after that, that will be multiplied. So that will enter into the seed production chain. So that will be multiplied into breeder seed, then foundation seed, then certificate seed will reach the farmers. So in this scheme, where we are doing the maintenance breeding, we are doing the maintenance breeding while we are multiplying the breeder seed. The aim of the maintenance breeding is maintaining the genetic purity of the breeder seed. So whatever the breeder seed which we are providing to any institute for further multiplication, that should be true to type of that particular variety. So our aim is maintaining the genetic purity of the breeder seed. For that, what we have to do, every time we have to produce the nucleus seed through maintenance breeding. Through the maintenance breeding, every time, every year, we have to produce the nucleus seed. From that nucleus seed, again, the breeder seed will be multiplied and that will go in seed production chain. So whatever seed which we are providing to the farmers, that should have some important quality attributes like genetic purity, physical purity, germination percentage, seed vigor, freedom from disease and insect. So among these qualities, genetic purity is important. It is very important, not only to realize the yield potential of that particular variety, but also it is important to maintain the particular variety in the field. So if you want to maintain the particular variety for a longer period, every time we need to maintain the genetic purity at the beginning stage. At the beginning stage means at the time of nucleus seed production itself, at the time of breeder seed production itself. So genetic purity is important both to realizing the yield potential of the particular variety, at the same time to maintain the particular variety for longer duration with its all original characters. So some of the factors, these things everybody knows very well. So these are all some of the character which is responsible for deterioration of any variety. When we are doing the maintenance breeding, we need to take care of these things. So these are all some of the factors. For example, once any breeder releases any variety, once if it come into cultivation or multiplication as well as cultivation, so there are so many factors. The one is developmental variation. Developmental variation means Wherever we are cultivating in some other places, at the time, some of the character which is not expressed at the time of developing the particular variety, that may express later time. So that may cause variation among the variety. So that is one reason. Another one is mechanical mixture. There is so many reasons are there, from the sowing, harvesting, processing, bagging, storage. In any stage, there is a possibility for mechanical mixture that may cause deterioration of that particular variety. Then mutation. Mutation is a problem, particularly in vegetable pea. Very less, it occurs in very less only. But when we consider the pea, when we are growing larger population, the deterioration will be maybe more due to mutation. Then natural crossing, because when we see the varieties, self-pollinated varieties are there, cross-pollinated, often cross-pollinated. So this natural crossing is a major problem in case of cross-pollinated varieties. Then genetic drifts. Sometimes we may lose some of the characters, some of the specific genes at the time of multiplication. Then selected influence of pest and disease, because one variety, if any breeder is releasing any variety, he may claim that that particular variety is having resistant to any virus. So what that virus will do, after within 1 or 2 years, 3 years, it will break the resistance of that particular variety. After that, the particular variety or hybrid may become susceptible to that virus. So we need to properly manage the pest and disease also at the time of our maintenance breeding. Then techniques of plant breeder. Plant breeder may use many techniques. So when he is using many techniques, at the time of release, the variety, the nucleus seed may not be a pure seed. It may be a segregating population. At the time, the seed producer may face a lot of problem. The maintenance breeding is very important to maintain the genetic purity of the breeder seed. If the genetic purity of the breeder seed is not maintained properly, what will happen in the next stage? Multiplication of foundation seed, multiplication of certified seed. At the time, we may need to put a lot of energy. In one stage, if you do mistake, at the time of maintenance, at the time of nucleus production, seed production, if you do any mistake, in the next stage, we need to put a lot of energy, lot of money. So that is a negative problem for us. So what we have to do, continuously, a breeder or any sponsored breeder or a breeder authorized by a main breeder or sometimes seed technologist, because when it comes to seed production, the breeder may not produce the seeds. Always the breeder may not produce the seeds. So the seed technologist, the authorized person in any institute, may involve the production of any particular variety or hybrid. So they should do maintenance breeding. Every time continuously they have to do maintenance breeding to maintain the original characteristic of any particular variety or hybrid. So this is our objective. What is the objective of maintenance breeding? Objective means here what we are doing, again and again we are producing the nucleus seed for the multiplication of breeder seed in maintenance breeding. This is a part of normal breeding procedure only, but here the selection should be mild. It should not be a strict selection. Here we are not improving the variety. In maintenance breeding, we are not improving the variety. We are maintaining the particular variety with its actual original potential. This is our objective of the maintenance breeding. So whoever involved in, because some of the photographs I want to share, whoever involved in maintenance breeding, you see, generally whatever the principles which we are following for seed production, all the procedure and principles we have to follow for maintenance breeding also. So the person who is involved in maintenance breeding should be very thorough with all the characteristics of particular variety. If you see this photograph, you see here, this is these are all two different varieties. In this variety, you can see only single flower per peduncle. Here you can see double flower, 2 flowers per peduncle. So here also you can see only single pod and double pod. When you see from a larger distance, you cannot identify, you cannot see the variation, this kind of variation among the plants. So the person who is involved should know all these characters before raising the crop. If you see here in the P itself, this is the original Azad P3 variety plant. This is the original plant. Here, what are all the variation which we can observe here? Here you can observe the yellow color pod. So here you can observe short internodes. Here you can observe there is no tendrils. So these kind of art types you can find in the field. So the person who is involved in maintenance breeding should know the characteristic and observe the plant closely. Here also the same thing. Here you see the peduncle, length of the peduncle in the pod. The length of the peduncle is here it is very small. This is the original character. Here, if you see the length of the peduncle is more. So this kind of variation one should know. Sometimes you can find the half type, you can observe the half type from the distance itself. The plant which is a lengthier plant, and sometimes you find in the field half type like flower variation. So this kind of variation you can find the length of the pod in case of cowpea. If you see here, you see these things. This is the radish plant. These both are two different varieties. One is Kashi Sweta, another one is Kashi Hans. The only difference is in the leaf characteristics. So this kind of variation like root shape, then core color. Here you see this is also carrot, Kashi Krishna carrot only. Here you see the core color. So this kind of half type has to be removed when any person involved in any seed production, not only in nuclear seed production maintenance breeding, any seed production, this kind of half type has to be removed. When we come to the hybrid seed production also, one should be very thorough with characteristic of male, female parents as well as hybrids. Because in hybrid seed production, both male and female parents are maintained separately up to the foundation seed level. When it's come to certified seed production, at the time we need to mate the cross and the hybrid seed will be distributed among the farmers. So these are all the things. But sometimes what will happen, we may commit some mistake. You see here, if the original characteristic is 2 flowers means when any person who is involved in monitoring, if he visit the later stage, this may fall down. This may fall down. He may find only single pod per peduncle. At the time, he should not judge that it is not true to type. He should not tell that it is not true to type, because these things are maybe due to because of some of the non-availability of nutrients, improper management. So these things should be avoided at the time of doing maintenance breeding. So this is also another problem. Some disease. Sometimes the later stage disease. Here this is yellow and mosaic virus. So the fruits become yellow color. The one should not judge that this is another variety. This may be another variety. Like that, one should not take any decision. So these things should be taken care at the time of producing nuclear seed, at the time of doing maintenance breeding. So so now we shall see the methods of maintenance breeding, because many methods are there. Whatever the variety or hybrids are there, it may be a self-pollinated crop or cross-pollinated crop. Accordingly, the methods will also vary, because among the plants, the variation, you can find more variation in cross-pollinated crops. So accordingly, the methods of maintenance breeding will vary. So now we shall see the maintenance breeding in self-pollinated vegetable crops. If you see here, generally from the nucleus seed, a handful of nucleus seed are already developed nucleus seed. From the nucleus seed, we shall raise the breeder seed production plot. So simultaneously, at the time of raising the breeder seed production plot, because here we need to follow all the measures like field standard, seed standard, whatever may be, we need to follow all the measures, isolation distance, monitoring from before at the time of vegetative stage, flowering stage. So all these things we carried out at the time of breeder seed production. So during that time, we have to select some of the promising plants. Among the plants, all the plants are true to type only, but among the plants, we have to select some of the plants showing true to type as well as high yield. So those plants we have to select from the breeder seed production plant. So those selected progenies will go into plant to row multiplication. The remaining whatever which we didn't selected, the remaining plants will be, the seeds from the remaining plants will be pulled together and used as a breeder seed, and it will go to further multiplication. So the seeds from selected plant we have to raise in single row. It may be a single row or paired row method. The length should be 3 meters. So we have to raise it in separately from plant to row. Individual plant should be raised in single row. Then we have to monitor closely regarding all the characteristics. So whichever rows which is showing the deviation from the actual characteristic, that has to be removed before flowering. But if you want to remove, if you are observing the variation or deviation after flowering or at the time of flowering, what you have to do, you have to remove at least the adjacent plant, at least up to 1 meter, or the maximum 3 meter in case of self-pollinated crop. You have to remove the adjacent plant also. You should not include the seeds from those plant in the nucleus seed. So what we have to do after removing these off type, we have to collect the seeds and pull it together. Again, we have to use it as a nucleus seed for further multiplication of breeder seed. So this is the simple scheme of maintenance breeding. Here we are our main aim is multiplication of nucleus seed only. Every time, this is our main aim. So if you see here, this is the plot. Here it is not properly visible. This is the plot. This is the copy plot. Here this is the breeder seed production plot. From here we have to select every year. Every year we select the plants, promising plants. We mark it and we select it. At the time of harvest, we harvest the plants separately. Separately we harvest the parts from the plant. Then we extract the seeds. Again we go for plant to row method of seed multiplication. So this is the one thing. The another thing is, in case of tomato kashi aman, this is one of the variety. Kashi aman is one of the variety which is having resistant to tomato leaf curl virus. So this kashi aman variety is having the tomato leaf curl virus. Here also we follow the same scheme, which whatever I have told till now, the same scheme only we follow. We select the plant from the breeder seed production plot. We take the plant, we select the plant, we harvest the fruits, we extract the seeds. The same procedure only we follow. Along with that same procedure, we confirm the presence of this TY3 gene. This particular TY3 gene is responsible for the resistant of this particular variety. The kashi aman is resistant to tomato leaf curl virus. This TY3 gene is especially responsible for this resistant. So each and every plant, whatever plant which we are selecting for the purpose of purification, we confirmed the presence of this TY3 gene by using this marker. This is the particular marker, particularly this is the gene specific marker P625. This is the gene specific marker. We used this marker, and this is the forward as well as reverse sequence of the primer. We used this marker. We confirmed the presence of this particular TY3 gene. You see 1, 2, 3, 4. These are all having particularly this TY3 gene. These are all showing heterozygous condition. These are all showing that 14, 16, 19, 35, 36, 29, 27. These are all showing heterozygous condition. This is also having the TY3 gene, but this is not pure. So we confirm the purity of the particular the selected plant through the molecular methods. Then whichever is not showing this heterozygosity, those plant only we will select for further multiplication of breeder seed. Here some variations is also there. Modification, not variation. This is modification. This is also same procedure only. But the modification is sometimes, for example, some of the seeds. In case of vegetable P, we need a larger quantity of seeds. Generally, we use at least 160 kg per hectare for the purpose of sowing. So vegetable P, we need a larger quantity of seeds. So at the time, a single stage of nucleus multiplication may not be enough. At the time, what we have to do? We have to go for the second stage of nucleus multiplication. So stage 1 as well as stage 2. We have to follow 2 stages for nucleus multiplication in case of P. If you see here, the up to this level, it is same. You are selecting the plants from the parental material and from the multiplication plant. From that plant, you are going to plant to row method of planting. Whatever showing half type, that you will discard. Only those plant which is showing true to type, the seeds will be collected from those plant, that is used as a nucleus seed, stage 1. Then next, what we have to do again, if we want to go to stage 2, if we need more nucleus seed, if we want to go to stage 2, what we have to do? We have to go to row to plant method. If you see here plant to row method. Now we have to go to row to plot method. So whatever the seeds collected from the plants in a particular row, that will be raised in plot. Then again we need to do the examination. If you find any deviation, all the plants which is present in the particular plot will be discarded. Those plants which is showing true to type in the remaining plot will be combined to seeds from these plants will be combined together and used as nucleus seed for further multiplication of breeder seed. You see, this is the plant to row method of seed multiplication in case of P, vegetable P. This is the stage 1 of seed multiplication. And if you see, if you want to go for stage 2, again row to plot method. First one is plant to row method, individual seeds from individual plant raised in a single row. Then after that, row to plot method. Though the seeds from the individual row will be planted together in a plot. Then you have to examine the plot. Then you have to harvest the nucleus seed. So this is also nucleus seed production stage 2. Then another one modification is there in case of P itself. There is another scheme also they are following. In this scheme, what they are doing, first they are selecting 150 single plant from the parental material. Wherever they are doing the multiplication plot, from that they are selecting 150 single plant. In the after selecting that 150 single plant, they have to examine all the 150 single plant. So we need the half type will be removed. Whatever the plants is showing half type, at least 20% of half type will be removed. Then remaining 120 best plants, we may find out remaining 120 best plants. So remaining 120 best plants, we have to select only 30 best individual plant progeny rows. So in the 30 best individual progeny rows, what we have to do? We have to harvest 10 best plant. For example, here 150 progeny rows. Here what we are doing, 150 single plant we are selecting. Then from the single plant we are going 150 progeny rows. Well, when after raising 150 progeny rows, among them we are selecting 120 best progeny rows. So in the 120 best progeny rows, again we are selecting the 30 best progeny rows. This is rows. So 30 best plant progeny rows. So from among the 30 best plant progeny rows, not among from all the 30 best plant progeny rows, we have to select 10 best plant. From the 30 best progeny rows, we have to select 10 best plant. Among the 10 best plant, again we have to select 5 best plant. So in the 30 plant progeny rows, if you are selecting 5 best plant, means we may get 5 into 30, 150 plants. We will get 150 best plant. So this 150 best plant will go again into the same cycle. This is the one part. Okay. We have selected 30 best plant progeny rows. What we are having? Remaining 90 plant progeny rows we are having. We are having the 90 plant progeny rows. We are having the remaining 90 plant progeny rows. Then again, among the we utilize this 30 best plant progeny rows also further multiplication. Okay, we use this 30 best plant progeny rows also for further multiplication. So this multiplied seed as well as the seeds from this 90 progeny rows will pulled together and used as nucleus seed for further multiplication. Here what we are doing, we are selecting 150 plant in one scheme for further multiplication for further round of selection. The remaining plants will be further multiplied into plot method that will also used as nucleus seed. That's all. So the cycle will continue. Nucleus seed production also will continue with another scheme in the same scheme. Then when we come to the cross-pollinated crop, here the problem is generally cross-pollinated crop is having showing lot of variation among the plants. This is unavoidable. So here what we need to do, we need to maintain large isolation distance. That is very important. Here we need to maintain systematically, we need to follow the procedure. That is one of the important thing. And also every time we can find the variation. In case of cross-pollinated crop, you cannot go strict elimination of the plant. In case of self-pollinated crop, you can consider the off type strictly. You can do the roguing and all those things. But in case of cross-pollinated crop, strict roguing cannot be possible because always you may find the variation among the plants. So here what we are doing, here we are doing the negative mass selection. A simple method only. One plot is there, that is breeder seed production plot. In that plot, one part we need to allocate for nucleus seed production. So what we should do, strict monitoring, strict evaluation, we shall do always in that particular plot, that nucleus seed production plot, and under the supervision of that particular breeder. That is also very important. Whenever we are separately, we are developing the nucleus seed in a particular plot, what we have to do, we have to invite the breeder. Under the supervision of breeder only, the person who is involved should carry out the roguing and all those things. So strict roguing will be carried out. Whatever the remaining plant which is showing the prototype, the seeds from the remaining plant will be pulled together. Those seeds will be used as a nucleus seed for further multiplication of breeder seed. Here only thing is we have to maintain the isolation distance and we have to follow the procedure properly. Otherwise, there is a possibility for cross contamination. This is one of the negative aspect of this negative mass selection. But the thing is that this is a quick method. In the breeder seed production part itself, a part will be separately allocated for nucleus seed multiplication. Strict roguing will be carried out. Then remaining seeds from the remaining plant will be pulled together. This is one thing. In case of melon, musk melon, and this watermelon, after doing the mass selection, the purity of that musk melon as well as watermelon that will be confirmed through TSS. So we need to test the TSS. If in case of watermelon, it should be at least 10%. In case of muskmelon, the TSS should be 12%. So after checking this TSS and also flesh color, fruit shape, all those things, we need to do the selection for nucleus seed multiplication production. The another method is rest seed method or reserved seed method. Rest seed method or reserved seed is very simple. What we are doing, we have to select at least 300 to 500 plants. So we have to select 300 to 500 plants. Among the 300 to 500 plants, again we have to select at least 200 best plant. So what we are doing, we have to collect the seeds from 200 best plant. So here we are not going to raise the entire seeds from all the plants. What we will do, from each plant, we keep some quantity of seeds in reserve. So very less quantity of seed will be raised in row. So what we shall do, the row will be examined. Then whichever row showing the variation, that will be discarded. Simultaneously, the part of the seed reserved, that will also be discarded based on the assessment in the field. So here what we have to do after that assessment, whatever the seed which we are having in our hand, that will be pulled together, that will be used as nucleus seed. Okay, this is the reserve method or rest seed method. You see the scheme here. This is the parental material. Here, this is the multiplication plot. So what we are doing here, 300 to 500 plants we are selecting. Among the 300 to 500 plants, again we are selecting the best 200 plants. So here what we are doing, a particular quantity of seeds from individual plant, we shall keep with us as a reserved seeds. So very less quantity of seed will be used for to raise the row, plant to row method. So here what we are doing all the examination. We are finding out the off type. We are removing the off type. At the same time, what we are doing, the seeds, which the reserved seeds of this particular off type will also discard. We will discard the reserved seeds of this particular off type also. The remaining seeds, reserved seeds, will be pulled together and used as nucleus seed for further multiplication of breeder seed. So at the same time, simultaneously, in the among the 200 best plant, what we have to do, progenies of among the 200 best plant progenies, again we have to select 300 to 500 best plants. We have to collect seeds from 300 to 500 best plants. So this again enter into the same cycle for further multiplication of nucleus seed. This is the another scheme of nucleus seed production and multiplication. Sometimes we may need to maintain the genetic maintenance breeding in polyhouse also, because some of the varieties, like here you see, this is the Indian spinach, beta vulgaris. This is having this is a wind pollinated crop. It is showing highly cross pollination. So in this kind of crop can be raised inside the polyhouse. Sometimes the parental materials of hybrid seeds, hybrid also can be raised inside the polyhouse. That will be separately multiplied. Then in case of root crop, what we have to do, the main thing is before planting, because we are doing in two stage. First we are developing the roots, preparing the steckling, then transplanting the steckling. So at the time of steckling preparation, we have to observe the roots critically, then that will be transplanted. Here what we have to do, here at least 250 true type roots selected from individual variety, that will be multiplied. Here the thing is early type, early bolting plant, as well as those who are those plants showing late bolting also, that has to be removed. This is one thing. So the from the seeds from the remaining plants is to be harvested, and that is bulked and that is used as a nucleus seed for further breeder seed multiplication. Here this is the root selection, multiplication, nucleus seed multiplication in isolated condition. Then in case of bulb crop also same, because this is also cross-pollinated crop. This is also same, just like root crop. Here we have to select 250 typical bulbs. We have to select, and that has to be isolated, raised in isolated condition. From that, the early bolter as well as late bolter will be removed. Then the seeds from the remaining plants pulled together and used as nucleus seed. This is the another scheme. Then the one thing I want to share with you, I told earlier also, in our tomato Kashi Aman seed purification, nucleus seed production, what we did, we used the molecular methods. We used the gene specific marker for the confirmation of the purity of that particular plant, selected plant. In that same way, what we have to do, we have to use the molecular methods also for the confirmation of genetic purity of that particular variety, so that we can confirm the purity of the particular variety. Because the main thing is nucleus seed is important. Once if we commit mistake at the time of production of nucleus seed, after that everything will become mistakes only. The one mistake will cause lot of take lot of energy from us, money from us, so loss from us. So in the beginning itself, what we have to do, we have to properly do the maintenance breeding. We have to take the support of molecular methods. We have to take the support of molecular methods, so that the only thing is the purity of the variety can be maintained for longer period. The another suggestion is some of the companies, some of the private companies are also there in our country. So they are also providing services, because we may think, because now the problem is nowadays we are not using the molecular method for this type of confirmation. Even for genetic purity to grow out test also, we are mainly we are doing grow out test, but not in the field only. We are not using the molecular methods for grow out test also. But the only thing is, the main reason was there is no trained person. There is it involved a lot of cost. Those things are there. So what we can do, we can use the service of this kind of people. So private companies are also there. So what our thing is, we have to provide the sample, we have to give the sequence information. So they will do all the work, they will confirm the purity, they will give the result. So we can use this kind of services which is available in the field also. So the main thing is, maintenance breeding means it involves purification of breeder seed, purification of production of nucleus seed, then production of pure breeder seed. That is the main aim in case of maintenance breeding. Thank you. If you
Thank you so much, Manimurugan sir, for giving such a nice presentation. With the
Okay.
permission of speaker, now the session is open for discussion. If any questions are there from participant side, means just unmute yourself and you can ask questions to the Manimurugan sir.
Thank you, sir, for. Hello, sir.
yes, sir. Please.
Thank you, sir. Myself, Dr. Sonata Das from OUT Bhubaneswar, sir.
Namaste,
Your
madam. Namaste.
presentation was very nice, sir. And actually, it is very nice. And sir, one thing I want to ask you, sir, how these molecular markers are used for maintenance breeding? That slide, tomato leaf curl virus slide, sir. If you can explain it again, it will be better.
Okay, madam. Just I shall. I shall. Should I share my screen?
Yes, sir. Just reshare it and you can proceed, sir.
Actually, I don't know how to.
Sir, just
tomato leaf curl virus.
share screen, sir.
That one.
Sir, first open your PPT, sir. First you open your. Just open it.
From my.
Yes, sir. From your side only. Just open your PPT and then go to the share screen, sir.
Okay. So is it visible?
Yes, sir. Yes, sir.
Oh, sorry. Actually, madam, this is the scheme, madam. For example, you consider this is a tomato breeder seed production field. Okay, here we are selecting the individual plant. In the breeder seed production plot itself, we are selecting the individual plant which is showing true to type, which is having higher yield. So we are selecting the individual plant. But all the plants are showing the resistant. All the plants, these plants are, you think that all the plants are showing resistant to virus. But what we have to do, we have to confirm whether the particular plant is having virus or not. That is one thing. Or is there any contamination in that particular plant?
Okay.
This is what our aim.
Okay,
So,
sir.
we have to confirm the presence of that particular gene. If you see here, this TY3 gene is a particular gene which is responsible for resistant of this particular variety.
Okay.
Against this tomato leaf curl virus. Because of this TY3 gene only give the resistance to this particular variety. What we have to do, we have to confirm whether the TY3 gene is available or not, whether it is present in the plant or not. That we need to confirm. So already we are having a marker. This is the marker which already developed by the breeder. P625 is a marker. This is specific to this particular gene. Only if this particular gene is there, then only you can find this amplification. If you see here plus as well as minus, this is the plus. Plus means this is the seed, the plant, the sample which is the breeder is having. This is the confirmed source of that particular TY3 gene. This is the negative means this is another variety, any other variety which is not having this particular TY3 gene. So this is plus minus means this is the plant which is having TY3 gene. This is the plant which is not having TY3 gene, and this is the test sample. These are all test sample. So among the test sample, if you see 1, 2, 3 up to 13, all the plants are showing amplification of this particular TY3 gene. It means this particular gene is available in all the plants. So in case of 14, 16, 19 also, you can see amplification also. You can see amplification for TY3 gene also. At the same time, this is further it is contaminated with some other pollen grain. So it is showing heterozygous condition. It is not homozygous. So what will happen in the next generation? It will segregate. It will segregate. You may find some of the plants which are showing what we can say susceptible to a particular virus in the next generation. So our aim is at the time of nuclear seed production itself, we have to eliminate this kind of plants. So this can be done only with the help of these molecular methods. Otherwise, it is not possible. We cannot confirm. We cannot confirm the heterozygosity, and we cannot eliminate this kind of plants. What through our naked eye, what we can see, all the plants are showing resistant. So all are having true to type. What we will do? We will do mistake. So again in our nuclear seed production plot, again we will find the segregation. Then the segregation will cause will show the susceptibility in the field. Then we will think that people told that this particular variety is very resistant. Now there is a break in resistant. This is not true to type. So like that we may think. To avoid those things only, we have to use this molecular method. We have to confirm the presence of that particular resistant gene. Then we can. So this is one confirmation method only. Additional support. Hello. Is it okay, madam? Hello. Hello.
Sir, can I ask a question?
Yes, sir.
Sir, myself Noor Nawaz from College of Agriculture, Hanumanmatti.
Okay, sir.
Sir, you told that in cross-pollinated crops, roguing removal of types and roguing is difficult. Why it is so, and if it is so, what are the other strategies to achieve?
Difficult in the sense we cannot go for strict roguing.
Yes, sir.
Strict roguing means strictly you cannot eliminate the plant because always you can find some of the variation because since it is a cross-pollinated crop.
Yes, sir.
You cannot get exactly true to type always.
All right.
Always,
yes,
in case of cross-pollinated crop, always you may find a little bit variation. So at the time of development itself, the breeder will give with that variation only. So at the time, it is the what you can say, the person who is doing the maintenance breeding should take care of all those things. If you do strict roguing, we may lose some of the characteristic of that particular variety. So that is the reason we should not do strict roguing in case of cross-pollinated crop.
Right, sir. Are there any other strategies, sir, to achieve?
No, not strategy. That is the method. This is the only method. Otherwise, if you go for strict roguing, just like genetic drift, we may lose some of the genes. We may lose some of the qualities. Then day by day, the variety will get deterioration. That is the reason.
Okay, sir. Thank you, sir.
Okay.
Hello, sir. This is Mantesha. Hello, sir.
Yes, please.
So is it audible?
Oh, wait. Some problem in my.
Sir,
yes,
just
it is audible.
Sir, unshare your screen, sir.
Unshare, huh?
Yes, sir.
That is.
Stop sharing, sir. Stop sharing.
Okay, stop. Yes, please. You please continue.
Sir, this is Mahantesha here.
Yes,
So
yes.
you have shown that using the molecular marker for genetic purity assessment,
Yes, yes.
so single gene specific to the disease you have expressed.
Yes, yes.
How
Resistant.
about if suppose multiple genes are responsibility for the single disease?
So in that case, the person whoever is developed that molecular marker, so he only can give proper guidance.
Okay, okay, sir.
Yes, yes. Because for that only if there is available in the available any marker, then only we can go.
Okay, okay,
If the
sir.
markers are not available, if it is not developed, it is not possible. We cannot do. The person who is involved in maintenance breeding cannot do these things.
And another thing, the marker which you have used, it is already very well confirmed that it is validated.
Yes,
And
yes,
it has
yes.
shown that this marker is known to involve in resistant for disease.
Yes, yes, yes.
So confirmed marker only you have used.
Confirmed marker only. That is very specific to that particular gene. That is confirmed by the breeder. Or the,
and validated.
Yeah, validated by the breeder. Yes, yes.
Yeah. Okay, sir. Thank you.
Thank you. Thank you very much.
Hello, Mani Murugan sir. This
Yes,
is
sir.
Dr. Sivaramakrishnan.
Oh, namaste, sir.
Namaste, sir. First of all, it is a very informative lecture, sir.
Oh, thank you, sir.
And we get lot of information regarding maintenance breeding.
Thank you, sir.
Sir, my question is regarding that hybrid purity testing, sir.
Okay, sir.
That in hybrid purity testing nowadays, during sampling, we are not sampling the individual plant. There is one bulk method of sampling.
Okay, sir.
So, sir, will you please give some information about bulk method of sampling, that how we will do that bulk method of sampling?
Sir, actually in hybrid seed production, generally we are maintaining the parent line separately, sir.
Yes.
Up to certified seed production, from whether it is nuclear seed production, breeder seed production, foundation seed production, whatever may be, we are maintaining the parent line separately. So when we are maintaining the parent line separately, in case of bulk method, what you are telling, so we have to go for negative selection only. Whatever showing the off type, that has to be removed. The remaining plant has to be pulled together and used for further multiplication, sir. So like that only, sir.
Okay. Okay, sir. Okay.
Okay, sir. Thank you,
sir. Hello, Mani Murugan, sir. Good
yes,
morning. I'm Chitra.
Good morning, madam.
Very good presentation. I am
madam.
from NPPGR Delhi. Sir, I would like to know, like, how do you maintain the plant germplasm, especially with respect to cucurbitaceous vegetable?
So that may be a part of plant breeding work, madam, but they are doing selfing only. Selfing only, male and female flowers covered separately. Then the next day, before anthesis, the next day we have to pollinate with the plant. But here also, in case of seed production, this is the problem in cucurbitaceous crops. In case of seed production, because after flowering, once fruit sets are there, after reaching a particular stage only we can differentiate the fruit shape from one fruit to another fruit. At the time, the half type may contaminate the entire field.
Yes.
So in
but
this,
we have to maintain the genetic purity of that particular material.
Cucurbits, madam.
Cucurbits.
So what we do, madam, in case of cucurbitaceous crop, we have to do the selfing only. We cover the both, especially this one, bottle
before
guard.
Okay.
Before flower opening, before the anthesis, we cover the male and female flowers. We do the selfing, madam. We do the selfing. So once the fruit sets are there, after that you may find some variation also among the fruit shape.
Yeah.
So at the time you need to discard the half type fruits. You have to retain the fruits which are showing prototype. So from that again we need to go for further multiplication, madam.
Okay.
We did like that only in case of cucurbitaceous crops.
Okay. Thank you.
Yes, madam. Thank you, madam. Thank you.
Hello, good morning, sir.
Good morning, sir.
Myself, Dr. B.K. Sinha from Sukash, Jammu.
Namaste, sir.
Sir, namaskar, sir. Sir, your talk is a very elaborative and very informative, sir.
Thank you, sir.
So I want to know, sir.
Yes, sir. Yes,
Hello.
sir. Please, sir.
I want to know what is the impact of climate on the seed production. Actually, all the seeds is not growing in all the areas. So
Okay, sir.
just like onion is a very costly seed. So
Okay,
can
okay,
we
sir.
grow in the northern part of the India?
Onion, sir.
Onion,
Yes, sir. Because we are doing seed
onion,
production in
carrot.
northern part of India.
G.
Yes, sir. We can actually the seed production of onion, seed production of nucleus, seed production of carrot. These are all carried out in this one IR regional station Karnal by Dr. V.K. Pandita. That is colder region only, so that can be cultivated, sir. The only thing is when we are going for hybrid seed production, at the time the climate may have a lot of impact. And also sometimes the temperature have a lot of impact in flowering also. In case of tomato, flowering may be there, but seed set may not be there. We faced that kind of problem during winter in our institute itself. Fruit
Can
set
we
also
grow in
there. Yes.
Jammu climate? Can we grow seed of the onion, carrot, and this in Jammu region?
Jammu region also you can try in this one, sir. Summer, sir. I don't think I don't know exactly. But
the maximum temperature was 45 or like that, and minimum is -2, -1.
-1, -2, it will be very difficult, sir.
Yeah.
Yes, sir.
It's difficult, no?
Yes,
That's
sir.
why
You can do that one in case of this one, sir. Controlled condition, polyhouse condition. You can do, sir.
Yeah.
Yes, sir.
Thank you very much, sir. Your
thank
talk
you, sir.
is very informative and elaborative. Thank you very much, sir.
Thank you, sir. Thank you, sir. Thank you, sir.
Is there any participant want to ask question?
Hello, sir. Myself, MS Patil, sir, from
Yes, sir.
Solapur, JDR Solapur.
Okay, namaste, sir.
Namaste, sir. Sir, in case of drumstick, how to maintain the this one, sir? Breeding lines.
Genetic purity, sir.
New variety, release variety. Drumstick, sir. This one.
Drumstick, actually, drumstick is highly cross-pollinated crop, sir.
Yes, sir. Then how to maintain the purity, sir? Genetic purity.
Sorry, if you want to maintain the genetic purity, you can go for cutting only, sir. Cutting, but generally, this one drumstick multiplied through cuttings.
Limbs, yes, sir. Limbs is there. But suppose I will release one variety. How to maintain the, suppose in case of dwarf plant, we can cover by the means of insect net and other things. But in case of tall trees, how to maintain the genetic purity?
Sir, either you can cover or you can go for vegetative propagation, sir. Yes, sir.
Hello.
Hello, sir. Hello.
Hello.
Yes,
good
sir.
afternoon, sir.
Good afternoon, sir.
Sir, thanks for nice talk, sir.
Okay,
and I am Dr. Harpal Singh from Punjab, sir.
Yes, sir.
Sir, I have a question regarding chickpea. Sir, is there any work going for this introduction of chickpea in case of arid zones, which we have disease-resistant variety?
Sir, actually, in case of chickpea, arid zone means you are
in Bathinda, sir. Sub-arid zone, sir.
With respect to chickpea, I don't have much idea, sir. But
okay, sir.
Yes, sir.
Okay, no problem, sir. Thank you.
Okay, sir. Thank you.
Sir, good afternoon, sir. Sir.
Yes, sir. Namaste, sir.
Sir, myself, Dr. Somappa Jaggal, sir. Sir.
Yes, sir. Yes, sir.
Sir, I am asking about how the maintenance breeding of pigeon pea, sir. Sir. Because pigeon pea having frequent flower dropping major drawback, sir.
Okay, sir. Actually, in case of pigeon pea, generally they cover the whole plant, sir.
Sir.
You cover the whole plant. By that way only you can go for genetic purity maintenance. Otherwise, that is also highly cross-pollinated crop, sir.
Because of honeybees problem, sir. No, sir.
So that you can do only covering the whole plant with a net.
Whole net, sir.
Yes, yes, sir. Yes, sir.
Very nice talk, sir. Very enjoyed, sir. Yes.
Thank you, sir. Thank you, sir.
Thank you very much, sir.
Thank you, sir.
Hello, good afternoon, sir.
Good afternoon, sir.
Sir, myself Paranjay, I am a PhD student.
Yes, yes, sir.
Sir, I want to ask if during any adverse climate conditions, say
Yes.
whole lot in the field, it shows a lot of variation. Then what we need to do, sir?
No, suddenly you cannot see that kind of variation. Due to adverse condition, what may happen? Sometimes we may lose the entire seed.
Yes, sir.
For that purpose, we have to always have some quantity of seed in reserve.
Okay, sir.
That is important.
Okay,
Sometimes this is the problem. Sometimes what will happen due to adverse condition, our entire field will be devastated. We may
Yes,
lose
sir.
all the seed multiplication plot.
Yes,
So for
sir.
that purpose, we always need to keep quantity of seeds in reserve.
Yes,
That
sir.
is important in all the case.
Yes, sir. In the same case may be applied to some biotic as well as abiotic stresses.
Biotic abiotic stress also, but that is depending upon your objective.
Okay.
Oh,
Thank you, sir. Thank you. It was a very nice presentation.
okay. Thank you. Thank you. Thank you, sir.
Okay, thank you so much, sir. If there are no questions from participants, it means we will wind up this session. Sir, thank you so much for presenting such a very informative and elaborative session, sir, as expressed by the participants. And also, I am very happy to share with the participants that me and sir were in the same batch during our PhD at IRI New Delhi. Sir is very hardworking and very humble person. Thank you, sir. Thank you so much for accepting our invitation and giving such a nice presentation. May I request Dr. Ganesh Gowda I Patel, sir, to propose vote of thanks. Over to you, sir.
Thank you, Dr. Dhananjay. Sir, thank you very much, Dr. Mani Murugan, sir, for accepting our request to give talk in this 10 days national level training program on this topic, an overview on nuclear seed production and maintenance breeding. So your presentation was very informative, very elaborative, and useful to all the participants, as they have asked many questions and you have cleared the doubts. So they are very much happy happy with your presentation, sir. So in this connection, I am on behalf of College of Horticulture, Bidar, and University of Horticultural Sciences, Bagalkot, and on behalf of organizing team and also participants, I am very much thankful to you, sir. So and also the participants, also we are very much thankful to them for their active participation in this training program by asking many questions and clearing the doubts and clarifications, and also hearing the talk very patiently. So I am very much thankful to both the presenter and also the participants and also the organizing team. Thank you very much, sir.
Thank you, sir.
Thank you so much, sir.
Thank you. Thank you.
The participants are requested to rejoin at 3 o'clock for the afternoon sessions.